we have moved on to our next chapter. Now, as we've gone from skeletal system to the joint chapter, which also has to do with skeletal system, it makes a little bit of a move from talking about bones as these organs that support and protect, etc., but also we know are required for movement. So the joints are where mostly movement's going to occur, right? Depends on the type of joint. And now we're going to talk about muscle. So muscle, at least skeletal muscle, is what's going to make those joints move. What's going to make that skeleton move. Now we'll find that there are 3 different types of muscle. So you'll need to know all 3 types and specific aspects about each, about their anatomy and their physiology. But then also, you'll find that we spend most of our time learning about skeletal muscles. So we're going to use skeletal muscle as kind of our template muscle. We're going to learn the most detail about that, and then towards the end of the chapter we'll do some comparisons of the other muscle types with skeletal. How are they similar? How are they different, both in terms of structure and in terms of function? So cardiac muscle we'll get to in Bio 202 as well, of course, with the cardiovascular system. Smooth muscle we'll see here and there. We saw it in the integumentary system, right, like the erector pili muscles. You'll see it in places like the eye, right, when the iris, where we can constrict or dilate the pupil. You'll see it definitely along the digestive system. So various places that it will crop up. So we'll see that comparatively later on in those systems, in those chapters, etc. All right, so let's take a look at starting with some random muscle facts, right? Those are always good things to impress your friends with. Muscle actually means little mouse. So in fact, the standard house mouse is, they have a binomial system for naming these different species, and it's called Mus musculus. So mus is its genus and musculus is its species. And so you can see how it relates to muscle. Once upon a time, before they got into the science of studying the human body by doing dissections and looking inside of it, they would see muscles move, and as those muscles move, they would think that they look like little mice running underneath the muscles. Now, of course, it depends on how big that muscle is, but that's where the name came from. All right, other things. About 700 skeletal muscles, or in the 600 range. It depends on the individual. There's a lot of variation there. That's substantially more than the 206 skeletal muscles, or skeletal bones that we found. But don't worry, we won't be learning all of those, just a subset to represent the system, to look at the particular muscles that move our body, that are primarily used in everyday life. Other things. Smallest skeletal muscle is called the stapedius. So that is a muscle in the middle ear. So the smallest bones in the body, like you may have read about, right? 206 bones, 3 here, 3 there. That's the 6 from the 206. There are 3 ear bones, the malleus, the incus, and the stapes in there, that basically receive the stimulus from the tympanic membrane, and they beat out that rhythm onto a part of the inner ear to transmit that signal. So the stapes is the smallest bone, and the stapedius is a skeletal muscle that it attaches to those bones, and it keeps them, when there's really loud music and they might be going all over the place, it dampens their movement so you don't get hurt. Other things, largest muscle, gluteus maximus, of course larger in some compared to others. Still the largest muscle. Most active muscle are the extrinsic or external eye muscles, because even when you sleep, those muscles are doing, like during rapid eye movement sleep, right, REM sleep, those muscles are also moving. Other things, all body muscles contracting in one direction would produce 25 tons of force. We'll find that that is not going to be able to occur, though, because the muscles are arranged on opposite sides of the limbs and the torso, etc., and they're going to produce opposite movements. So, can't really generate that, but interesting to think about. And banging your head on a wall uses about 150 calories per hour. Better ways to burn calories, etc., but sometimes you may use that during the semester. Okay, so when we look at this muscle tissue, we're going to start small and we're going to work our way up. So we'll get down to the cellular level, even down to the molecular level, in order to understand how these muscles do their job. They have one job: they contract. So when you stimulate them, they contract, and when you stop stimulating them, they relax. but they do so in different ways, using different proteins and things like that. So it's still nearly half of the body's mass. What they do is they transform the energy, the chemical energy in the form of ATP, into mechanical energy so that we can produce movement. So these are the various things that we'll be looking over coming up, right? We're going to look at types of muscle, and we're going to look at the characteristics of each type, and then muscle functions. So particularly when we get into skeletal muscle, and we're talking about what they move, etc., where they attach, that's when we get into some of the details there. So some of the things to keep an eye out for as you're going along particular prefixes and things like that, you can see how there is myo and mes and sarco. So those are going to come again and again, and what we find is that the myo part is one of the most common ones that we'll see. Like later on when we're talking about the reproductive system, there's a muscular layer of the uterus that's called the myometrium, things like that. Okay, so we'll see that coming up. 3 different types of muscle tissue. So we have skeletal muscle tissue, we have cardiac, and smooth, right? Those are the 3 types that we have. Now, and when we look at these, skeletal muscle is going to be our main focus. Now, only the skeletal and smooth muscles are really elongated and referred to as muscle fibers. So an individual cell is the same as a muscle fiber. So keep that in mind. All right, so skeletal muscle itself, packaged into the skeletal muscles, of course. So remember, each muscle counts as an organ. So because it's more than one tissue type, it's going to be mostly muscle, but there's going to be connective tissue in there. We'll talk about that. It's going to be nervous tissue stimulating it, even some epithelial tissue, some wrapping, right? So when we look at the skeletal muscle fibers in terms of their characteristic, they're going to be the longest. When we see them in the microscope, they're going to go so far beyond what we can see in that viewable area because they're so large. In fact, they're multinucleate cells, and they have striations or stripes on them. That's going to be a key characteristic to see and to help you identify those. They're also called voluntary muscle. Now, as you would imagine, right, when you move your eyebrows or move your tongue or something like that, or move your digits, right, that's all skeletal muscle, voluntary. Your mind is telling those particular muscles to contract and relax. It seems like second nature, like you're doing it kind of under the radar, but you're definitely doing it consciously. Cardiac muscle and smooth muscle tends to be involuntary instead. You don't have to think about it to make it happen. Okay. Contract rapidly, tire easily, but powerful. So as you can imagine, cardiac muscle and smooth muscle, we can't have it tire out, right? You can't get tired of digesting your food or pumping your blood, etc. So definitely some comparisons there. So we'll see some of these terms coming up again and again, but these are some of the characteristics, right? Skeletal, that it moves the skeleton; striated, that it's striped; and voluntary, that we have the conscious control over it from the brain. So cardiac muscle, just as a tour, found only in the heart. That's where the name comes from, right? Sometimes people get confused. They think cardiovascular system, that means we'll find that we have arteries and veins and arterioles and venules that all have muscle along them that allow them to constrict and dilate. But the muscle that's in arteries and veins is actually smooth muscle. Cardiac muscle only takes place or is occurring in the heart itself. It's also striated, so we can't use that one striation as a characteristic for only our skeletal muscle. Involuntary, like we said, that we don't have to think about it to make it do its job. Now, of course, the interesting thing is you can increase or decrease it voluntarily, right? You chant your mantra or something like that. Think of your happy place. Think of an exam coming up, and that can increase or decrease your heart rate, to be sure. But for the most part, involuntary. All right, smooth muscle then. So smooth muscle, like we said, is going to be found lining a lot of these organs that we don't have conscious control over. A lot of them are hollow organs, not all of them. Like we said, the iris of the eye, etc., the erector pili muscle. But a lot of the hollow organs that we have are going to be lined with smooth muscle, like the stomach and the colon and things like that. Even the urinary bladder and things like the airways, like the bronchioles, etc. Not striated, and that's where its name comes from, actually. It comes from the fact that it looks smooth in the microscope. And like we said, involuntary. So what we find is that, again, you can have the nervous system stimulating it, etc., and it's going to do so under the radar, It'll be coordinated, to be sure. It's not just randomly contracting, but it's going to be unconsciously controlled. So this figure from table 9.3 does a good job of giving you examples, showing you what they look like, and talking about their characteristics. So make sure you take a look at that. Okay, so regardless of the 3 different types, we're going to have 4 main characteristics for these different muscle types. They are excitable, meaning that we poke them, bzz, 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 electrically generally, electrochemically, they're going to respond. Now, excitability, we're going to find that that's also in the nervous system, so don't think that it's exclusive to the muscular system. Contractility, the fact that they'll shorten. Extensibility, that they can stretch. And elasticity, that they can go back to their particular resting length. That's going to be important as well. We don't want them all stretched out. For important functions, of course, producing movement, for things like locomotion, as well as moving things around the body, right? Moving the body as well as moving things around the body. Maintain posture and body position. Stabilizing the joints, like we saw. We talked about like the shoulder joint and how that cavity, that glenoid cavity, is not very deep, so we have a variety of tendons and ligaments to help hold it in place. Generate heat as they contract, because when we convert that chemical ATP into mechanical energy, it's not 100% efficient, so we lose some of that as heat, and that's good for us because we generate our own heat, right? We're endothermic, and so we need to be able to utilize that heat as well. So those are our 4 main functions, but of course there are some other ones as well in terms of protection and even forming valves and things like that, causing goosebumps. That's like we talked about, like the erector pili muscle. Okay, so let's take a look at the skeletal muscle anatomy, right? So we said it's an organ because it's made up of different tissue types. This is a key figure here, and like we said, we're going to zoom down to a microscopic level. So at the macroscopic level, we see that our muscle is attached to a bone, right? What bone do you think that is? Has a big head like that, goes into a big socket, right? Ball and socket joint. So that's a femur. Now when we look at that, is that anterior or posterior? It's hard to tell. We usually look towards the distal end, etc. Regardless, we're just doing a little exercise there. But we see the bone, and we see that the muscle itself is attached to the bone via this dense regular connective tissue that makes up a tendon, right? So the tendons attach muscles to bone. Ligaments attach bone to bone. All right, so we see our whole bone here. Looks like a piece of meat, right? You go to the meat department, guess what that is? All of that is muscle. That's what you're looking at. There's some interesting cuts as well, like steaks and things like that. If you look at some of the diagrams and you look at kind of the butcher's area, you can start to recognize where some of these different cuts are coming from and the muscles that you are looking at. So for instance, drumstick, right? Drumstick, chicken. You're going to find that drumstick is the leg, right? You asked for a leg as opposed to a thigh, right? We talked about the specific aspect of that. So the drumstick is the leg. The primary, the largest muscle on the leg, so on the posterior side, it's called the gastrocnemius and the soleus, right? That's the biggest part that you are eating, and it attaches via this Achilles tendon or the calcaneal tendon. So you'll start to recognize these things. T-bone steaks. T-bone steaks involve that T part of the bone. That's one of the vertebrae. So take a closer look at it. You'll see the body of the vertebrae. You'll see the transverse processes. You'll see the spinous process. Now, they have a T, so it's not the whole thing. They have it cut in half. So take a look next time you're there. Anyway, whole muscle that we see here is surrounded by a connective tissue layer that's called the epimysium. So see the MYS? That's the mesium part. Now, making up that whole muscle are these bundles of muscle fibers. So these bundles of muscle fibers, oh, my drawing skills or my highlighting skills are not very good right now. My pen isn't hooked up. There we go. So those bundles of muscle fibers are called fascicles. So the fascicles, and we're showing one coming out here, the fascicle is a bundle of muscle fibers. And then when we look inside of it, of course, we have individual muscle fibers. So this one thing right there is going to be a muscle fiber. So the muscle fiber is, what is it? Muscle cell, right? So the muscle cell, that's it. So a fascicle is a whole bunch of muscle cells. Now take a look. Wrapping up a fascicle is called the perimysium, and wrapping the individual muscle cells is called the endomysium, right? Endo is on the inside. So it's like those Russian dolls, right? from the whole muscle to the fascicle to the individual muscle cell. But it doesn't stop there. Oh, you might be excited by that, and you might be fearful of that. We'll see. Okay, so we're going to be zooming down to those levels. So anyway, we saw that skeletal muscle, it's going to attach to the skeletal system, it's going to span the joints and attach to the bones, and it's going to move those particular joints. Now, it's going to be attached in two different places. One's called the insertion, and we mentioned this a little bit before, right? The insertion is where we're attached to the bone that's going to be moving. The origin is where it is attached to the less movable bone, right? Origin is going to be immovable. Insertion is going to be movable. All right. So when we look at them, the attachments themselves can be direct or indirect. It all kind of depends on the type of muscle, the shape of it, the ligament or the tendon shape, etc. So the direct is where we see the epimysium, right? And that tendon going right into the periosteum, right? The periosteum is that layer on the outside of the bone instead. Or there's indirect. So indirect is where we extend beyond the muscle, like the tendon or even an aponeurosis. So, the direct is right into the bone, and then indirect, we have this dense regular connective tissue that forms a tendon. Or an aponeurosis is a big, broad, flat tendon, like over the top of the skull as well. All right. But regardless, we have to attach to the bones in order to move them. So Table 9.1 shows us that organization from muscle to fascicle down to cell. But notice, there we get a peek at our smallest level as well, right? We get down to the myofibrils. This is inside of the muscle cell. So we're going to be taking a look deeper into that. Okay, so let's look at that muscle fiber. That's where we're going to be focusing our time. So remember, the prefix sarco is also going to be showing up again and again, right? So these are very specialized cells, right? It's not just a generic round cell, so they have specialized names. So instead of the plasma membrane or the cell membrane, we have the sarcolemma. Instead of the cytoplasm, we have the sarcoplasm. And when we look at it, what we also find is that there's a variety of other specialized structures as well. There may be these things that are called glycosomes, which hold glycogen, which glycogen is a storage form of glucose. And then myoglobin, again, we see the myo prefix there, which is a protein that's able to hold and store oxygen locally. Other things that we find inside there, like we said, the myofibrils. We have a sarcoplasmic reticulum. Now, when we look at that, you're like, sarcoplasmic reticulum. What does that remind you of? Endoplasmic reticulum, just a specialized term. And then we also have these things that are called T-tubules. T-tubules, because what we're going to find is that when a muscle gets stimulated, it's stimulated right at the surface, and then we need that stimulation to get down in amongst all of these different myofibrils to tell them to move as well. So these T-tubules help to extend that signal down inside. All right, so let's take a look then. Getting into the inside, there is our sarcolemma, right? On the outside. Now we see our myofibrils on the inside. We see little mitochondria in between. We see nuclei. Remember, these are multinucleate cells. That's also one of our characteristics of skeletal muscle compared to smooth or cardiac. And we start to see those stripes or striation going up and down, right? So you can see them like this. So this is what it's going to look like in the microscope itself. So the striations, like we saw, goes dark, light, dark, light. The dark regions are called A bands, and the lighter regions are called I bands. And if you look at the vowels there, dark and light, that often helps us to remember A bands and I bands. There are also these things called H zones and M lines and Z discs. Let's take a look at all this that we're talking about. Now here's a key thing here, okay? The functional unit of the muscle is not the individual muscle cell. This is inside of the myofibril itself, and this is a contractile unit. It's called a sarcomere. So the sarcomere is the functional unit of the muscle. Important for you to know, that's where all the activity is going on to make the muscle contract. So you can see a sarcomere basically goes from one Z disc to the next Z disc, right? And it's called a Z disc because it has a zigzag, 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 etc. All right, so we see the A band here in the center of the sarcomere. We see I bands on either side. Now the Z disc, dividing one sarcomere for another, goes right through an I band. But that's our light, and there's our dark bands. And the reason that we have dark bands there is that we have overlapping filaments, what are called myofilaments. So these are inside of the myofibril. And these, when they overlap, they form an excessively dark area because they're so densely packed. Meanwhile, those areas where you see there's just these little light blue filaments, these thin ones, they look a little bit lighter. So imagine if you were looking at the page of a textbook from very far away, right? It would look the same. You'd have dark band and then light band and then dark band and light band. And the dark bands are where we have these densely packed lines of text, okay? That's kind of what we get in terms of the A band and the I band. The H zone is right here in the center. So we said that we have overlapping filaments through here. So there are thin filaments like we see here in blue, and then there are thick filaments which are shown kind of in the reddish color across here, okay? So notice that right in the very center of the A band, there's an area where we don't have any thin filaments, where it's just thick filaments. It still looks dark because the thick filaments, as their name indicates, are fairly thick, but we'll talk about that H zone later on as well. And M lines, we'll bring that in later on. Now this gives us a closer look at an individual sarcomere, right? From Z line, like we see here, to Z line. We can see in the light area, in here, it's just thin filaments, and now we can see that there's a term that goes along with it, and thin filaments are called actin filaments. And then these ones, these pink ones in the center here, these are the thick filaments, and you can see they have little extensions coming off of them. These are called myosin filaments instead. Now you can see these are both contractile proteins. We're talking about other types of proteins that are in there. You can see that there are elastic filaments called titin. So when we are pulling on everything and shortening the muscle, it gives us some of that elasticity, right? So when we relax and everything goes back to its original shape. So we do see some of those as well. But you can see, right, if you look at the edge of an A band, this is looking kind of end on in cross section. You can see both thick and thin filaments there, as opposed to the I band, like we see here, where it's only thin filaments shown in blue. Now here's a zoomed up version of our thick filament. Again, we see the little extensions coming off. So thick filament, we said, is called myosin, and each myosin molecule looks kind of like a golf club, right? So you can see there's a head to it over on the left-hand side over here, and then a long tail. And then, imagine, if you will, if a myosin molecule is an individual golf club, then if you take all those golf clubs and you put them in a golf bag and all those heads are sticking different ways, that's kind of what a thick filament looks like. Now the thin filament, interestingly enough, is a little bit more complex. So you see this blue layer in here, right? It's like a string of pearls. Those are the actin molecules. So these are the actin subunits. And so those are strung together like, maybe like a string of pearls, right? But notice it's not just the actin subunits. We also have these other ones that are called troponin and tropomyosin. So tropomyosin is that brown thread you see going around, and troponin is a three-part molecule you see here in yellow that occurs every once in a while. And the troponin and tropomyosin are called regulatory proteins instead. So actin and myosin are actually doing the contraction, but tropomyosin and troponin are controlling it, managing it instead. So they're associated with the thin filament. And this is kind of what it looks like when we look inside of the microscope itself. So you can see some of the myosin heads, you can see the thin filaments, so the thick filaments, and then the thin filaments are in between. So thick and thin and thick and thin. So this is our overlapping area of the A band itself. Okay? Okay, just a couple more things to look at. Remember we talked about the endoplasmic reticulum, the specialized one. It's called sarcoplasmic reticulum. So endoplasmic reticulum in a regular cell, right, makes proteins. If it's smooth, it makes fats, lipids, or whatever. And there is certainly some of that type of function in the sarcoplasmic reticulum, but one of the key things it does is it stores and releases calcium inside of the cell, intracellularly. Because calcium is a second messenger, and it's going to basically flood into the cells and tell the cells and all their myofilaments when it's okay to contract. We also have those T-tubules like we talked about. That are these invaginations of the plasma membrane. We call the plasma membrane the sarcolemma. And so they are going to go deep inside of the cell so that we can get that signal from the surface way up here down in amongst all of these myofibrils. Okay, so what we've done so far is we have given us the broad view. What does muscle do? We talked about 3 different types. We got to know the 3 different types, be able to discriminate between them. Now we're looking at the structure, the anatomy of the individual skeletal muscle cells, etc., because we said we're using skeletal muscle as a template. And now that we know some of these parts, we're going to start talking about how they work together in order to contract under what's called the sliding filament theory or the sliding filament mechanism. So that's next on our docket. So take a breather right now, allow that to sink in, get all those different levels going and stuff like that. Learn the vocabulary for the different names, right? Sarcolemma, sarcoplasmic reticulum, all that type of thing. And then be ready to talk about how those things interact to shorten your muscles.